Okay, I'm going to talk about the two main theories of color vision, how color vision works, and then I'm going to say this is how it actually is working for us. The first is the trichromatic theory of color vision, and that is how it looks and sounds. So tri meaning three, chromatic, chroma meaning colors, so that we have three color mechanisms from the trichromatic theory. This is sometimes called the Young-Helmholtz theory of color vision. Hermann von Helmholtz did a lot to systematize looking at this, and he was the one who really wrote up the theory. But the theory, trichromatic theory of color vision, really came out almost 50 years earlier than when Helmholtz was talking about it, and it was proposed by a Thomas Young, but Helmholtz ends up getting a lot of the credit for it, partly because of being famous, but partly because of the way he wrote it up. And this says, again, there are three basic color mechanisms, the trichroma, three colors. There are three basic color mechanisms, and the way they told this, or how they came up with this theory back before we really had very much physiological evidence, we're talking really about the late 1700s, early 1800s, was that they looked at color matching studies, and they looked at these metamers, where metamers are perceptually identical, so they appear the same when we're perceiving them, but they're physically different. The physics of what's out in the environment is different, and I'm going to show an example which hopefully brings that to where it makes some sense. So this is an example of a color matching experiment where they have a test field, and I'm going to have the example of 500 nanometers, which is a nice clear green. Okay, and that would be, they would have a light, and that would be a monochromatic light. Okay, so just 500 nanometers. And then they would give people, let's say, they would start with something like 420 nanometers, and people could adjust that light. How much light do I allow? I can make it lighter, brighter, or less bright. And with 420 nanometers by itself, obviously, that's going to never become green. That's always going to be blue to somebody who has three cones. They're going to be saying this. There's no way I can make green. Okay, they can give them then, I'm going to start with the 640 nanometers. So remember, this is going to be a red light. And if I add that to the 420 nanometers, can I get to green? Well, maybe I can get to green. But if I have two wavelengths that I am adjusting in the comparison field, I cannot create every single possible single monochromatic light from the test field. And so they didn't just test 500 nanometers, right? They tested several different wavelengths, and moving up, if I remember correctly, moving up by 10 nanometers or so, the next monochromatic light, and the next, and then the next. And so with two of the different lights and being able to adjust them in the comparison field, they could match some of those in the test field, but they could not match all of them with two. Then they gave them three. So now we're going to add the 560 nanometers, which is a kind of a yellow-green, or really a yellow. And now with three of those, as people were adjusting the lights, they could not only create the 500 nanometer green, but they could create any single wavelength that the researchers gave them in the test field. They could match in the comparison field with three wavelengths to manipulate. And so this is, again, an example of metamers, because what's in the test field is physically 500 nanometers, and what's in the comparison field is made up of three different wavelengths, but we can get it to be perceptually identical to what's in the test field. And because they could do this with three, for none of these did they need four. They could do it with four, but they didn't need four. They only needed three to be able to match any single wavelength in the test field, which suggests three color mechanisms to our researchers, or to our theorists. Okay, and I'm going to provide the physiological evidence. So that color matching experiment was really good behavioral evidence. And notice that those people were theorizing about this and doing those behavioral studies in the 1800s, 1840s at the latest. And this physiological evidence did not come along until the 1960s, so really over 100 years later, were we able to say, yes, we have physiological evidence for this as well. Those were assumptions based on behavioral support. In the 1960s, researchers measured absorption spectra of visual pigments in the photoreceptors. So they found pigments that respond maximally at 419 nanometers, so the short wavelength cones. And again, they respond to some extent around that, but maximally at 419 nanometers. They found pigments that respond maximally to 531 nanometers, the medium wavelength cones, and they found pigments that respond maximally to 558 nanometers, so the long wavelength cones. And so they found 3 different kinds of cones that each had their own absorption spectra. Later, researchers found genetic differences for coding the proteins for these 3 pigments, so in the 1980s. And if you read the Genes for Color Vision article, that is really about that discovery. Are 3 mechanisms necessary for color vision? Yes, for our range of colors and what we see, we need 3 cones or 3 color mechanisms, but no. We could see color with just 2. We're just going to be color deficient, right? We're going to be missing some of what a quote normal person sees. And I'm going to explain why, and then I'm going to show some of the examples of what other color deficient people would be experiencing depending on which cone they're missing. So I'm going to start with what if we only had 1 cone, because I've been talking about the ratios of these 3 different cones, but I want this to make sense to you. And so I'm going to start with, we're going to say J has 1 photopigment, okay? And his 1 photopigment, his 1 cone, it responds maximally at 550 nanometers, where it absorbs 10% of whatever's reflected into the eye at 550 nanometers of that wavelength. And 5% of whatever's reflected into the eye of anything that's 590 nanometers, okay? And we see those kinds of absorption spectra, right, in our photopigments. So if J is looking at Mary and Barbara, and Mary and Barbara have shopped at a monochromatic dress store where their dresses are indeed just 1 wavelength. Again, we really don't see that in the world, but it is something we could create. And if the light's shining on them that is causing 1,000 units of light to be reflected off the dress, okay, just 1,000 units of light is being reflected, and J's photopigment is absorbing 10% of the 550 nanometer dress that Mary is wearing, his photopigment is going to fire, basically, at, not really, but we're going to say that, fire at 100 units that quickly. Okay, he's looking at Barbara, who's wearing her monochromatic dress of 590 nanometers, and she's under the same light. 1,000 units are coming off of, are being reflected off of her dress of 590 nanometers. And now J's photopigment is only absorbing 5% of that, right? So now his photopigment is firing, or his cone is firing at 50. And it appears that Mary and Barbara have some difference there, and he can tell the difference. But all we have to do is change the light level. So now it's 2,000 units that are being reflected off of Barbara if we put her under a different light. And if we look at J's photopigment, it is still absorbing 5% of that 2,000, and now it's firing at 100. Well, now Barbara's dress looks exactly the same as Mary's dress, and now we can see why people with only 1 cone or with no cones are really getting shades of gray, because there is no distinction between these colors or these wavelengths. Now I'm going to go through and say, what if we had 2? Now we have 2 cones. So we're going to remember that we had 1 cone before. Now we're going to have Bob, and he has 2 cones. He has the same photopigment 1 that Jay had. He also has photopigment 2. And photopigment 2, it absorbs 5% of 550 nanometer light and 1% of the 590 nanometer light. And now we're looking at Mary and Barbara again. And when she's reflecting off 1,000 units of her 550 nanometer dress, Mary, the first photopigment is firing at 100, and the second photopigment is getting that 5% of this 550 nanometers, and it's firing at 50. We move over to Barbara under that where she's reflecting off 1,000 units of her 590 nanometer dress, and that first photopigment is again absorbing 5%, so it's responding at a level of 50 or 50 units, and the second photopigment is now firing at 10. And again, those appear different, but we have to change the light levels and see, does this really work for us to see these different colors, or for Bob to see these different colors. And so now we put Barbara under a light where she's reflecting off 2,000 units of her 590 nanometer dress, and let's see how our photopigment. So again, photopigment 1 is going to look just like it did on the previous slide. It's firing at a rate of 100 units. And that looks just like photopigment 1 for Mary. But if we look at photopigment 2 now, it's firing at a rate of 20, because now we're getting 1% of that 2,000 units of 590 nanometers, which is 20. And so now if we compare what's going on with Mary under 1,000 lumens and Barbara under 2,000, we see different ratios, right, between 1 and 2 for B and D there. We see different ratios between the cone 1 and cone 2, or photopigment 1 and photopigment 2. And so now we can tell the difference between Mary and Barbara. And with 2 photopigments, we can tell the difference between various colors or wavelengths, but there are some we cannot tell the difference between, because we have 3 cones. That allows us even more ratios to distinguish these colors with, or these wavelengths. Okay, so I'm going to talk about inherited color deficits as part of what this really tells us about trichromatic color vision theory, but also because color deficiency is interesting to most people, and a lot of people do experience this, where photopigments are either defective. So usually what's happening there is they're overlapping, the medium and long wavelength cones are overlapping more than they normally do in a quote normal person, and so we're not distinguishing colors as well. Or we have a photopigment that's just absent. And there are a number of different types of color deficiency, and I'm going to talk about really the ones where we have an absent photopigment. But you can think of it as potentially defective as well, in a number of different ways. And we determine color deficiency by color vision tests using Ishihara plates. And if you see, so if you're not color deficient, and usually we play this game in class where I say, what do you perceive an A? And most people say 74. There's a number 74. And we actually used an Ishihara plate when we talked about the Gestalt principles of the Gestalt principle of similarity, because we're grouping together, right, the greens, and we're grouping together, separating the greens from the reds, but grouping together the reds and oranges, because we're putting together things that are similar and separating them from things that are different. But some people do lack this distinction, especially the red-green distinction is probably one of the most common. And so when I ask, most of my class has been quote normal or had no color deficiency. When I ask people what they see in B, there is some question. And some people say 21, and some people say 71, and some people say 24. And I'm going to tell you that it's 74. This is actually the same exact Ishihara plate that is in A, except it's been manipulated to show us what a person who is color deficient is seeing. I have had several people in my class who are color deficient over the years, and they say A and B look the same to them, and when I ask them what's in A, they say it looks like, is it a 21? Is it a 71? I don't know. Is it a 24? I can't tell. It's very hard to see, as they are seeing what we are seeing in B. And trust me, that works, because I've had that, we've talked about it in class with people who are color deficient and are experiencing that. Some of this I'm going to tell you, there's no way for us to know what they're perceiving, because they're perceiving the world differently than us. And when we talk about it, we're just talking about what we've learned to call things. So anyway, or another way they've determined this is by color matching. So if you want to go back to where we looked at those metamers before, those people who are color deficient can match anything in the test field by using just 2 wavelengths and manipulating those in the comparison field. And I might say, as a person who does not have color deficiency, wait a minute, that doesn't match, but to them it does, and they only need 2. So in inherited color deficits, the photopigments are defective or absent, and usually what we see is dichromats. So this is on the X chromosome, and so if I'm a female, I'm getting 2 X's, and it's a recessive gene, and so I'm not very likely to show a color deficiency. Whereas if I'm a male and I'm getting an X and a Y, that Y chromosome is not carrying a lot of genetic material. And so I get this recessive gene, but I have the Y chromosome, and I'm much more likely then to show color deficiency. As a female, I'm just as likely to be a carrier, but I'm not likely to show the color deficiency. I need both of my X's. I need 2. I need a male parent who is color deficient and a female parent who is either a carrier or color deficient herself. And so they're missing a single photopigment in this case. Monochromats, so people who are really just seeing shades of gray, this is much more rare. We still have photopigments that are defective or absent. They might be missing 2 photopigments or missing all of their cones altogether. This happens only about 10 out of every 1 million people are monochromats. And even if they're missing 2 of their photopigments, but especially if they're missing all of their cones, they have poor visual acuity. They don't have that cone-rich fovea that gives us our visual acuity. They're very sensitive to bright lights. They don't see very well in bright lights, as we have talked about how our rods take a while to regenerate, and so we don't use them with our daylight vision, and so these people are just sensitive to bright lights, and their vision is based on the rod system. So I'm going to go through these next 4 slides, I think it is, that have these pictures that show us what a, quote, normal person sees over on the left, on your left, and then on the right is what the person who has this particular color deficiency is likely to see, or so we think. This is an estimate of what we think they're seeing. So people who have protanopia are missing their long wavelength cones. This happens to about 1% of males and about 0.02% of females. As we saw, this is one of the ones on the X chromosome. And you can see that they're really missing the red-green distinction, and they are lacking quite a bit in the yellows and reds. In deuteranopia, they are missing their medium wavelength cone. This happens again to about 1% of males and 0.01% this time of females. And if I usually do pop these back and forth and show how very similar the right example is in the protanopia and deuteranopia, because we're missing the distinctions. Trichromatic is more rare, so 0.002% of males and 0.001% of females have trichromatic, and you can see that they're really missing the blue-red distinction, and that's they're missing, we think, the short wavelength cones. There are fewer of these people. We have fewer short wavelength cones, and so this is what we think is going on with trichronopes. And the monochromat, either missing 2 photopigments or all cones, again 10 in about 1 million people, so this is very rare, and you can see it's really shades of gray in their experience. You can flip back. I mean, I usually flip around so that you can see the differences, and you can start over if you want, but it's hard for me to do that with creating these as videos. Just some fun cross-species comparisons, as I really enjoy kind of knowing what my furry friends are experiencing. Not always furry, but my friends are experiencing. Cats, actually dogs and cats and squirrels are dichromats, as many mammals are. Monkeys, not all monkeys, but New World monkeys and goldfish are trichromats as just examples. And then honeybees and birds are tetrachromats. They have more than 3 cones. If I remember correctly, it's 4. A couple of my students have written about shrimp that I think has 125 different cones, but they're in different parts of their different eyes that are experiencing different parts of their world. But it's fascinating. And it's just, I'm going to put this in here that we do appear to have some tetrachromatic humans. They are females, and they are getting some different genetics from, it appears, their color deficient fathers. They're not color deficient themselves, but because they have these different genetics, they can end out with, so usually, often what happens is you're missing one of those cones completely, but those cones can be defective to where they're not working, but they can be defective to where they're overlapping with another cone so much. That's actually called an anomalous trichromat, but so much that they're not experiencing the same visual world that we're experiencing. And these women can have what they get from their mom, their short, medium, and long wavelength cone, but then they also have this extra cone that is overlapping with one of either the medium or the long quite a bit, and they do distinguish more colors than those of us who are trichromats. There is some question of whether or not they exist, but it really appears that they exist, and it makes sense genetically why they exist. Okay, and there are still questions and phenomena that trichromatic theory of color vision cannot answer. And so someone, Ewald Hering, is going to argue with Helmholtz and Young for about 100, 120 years before we get that physiological evidence that we need to say which one of these theories is correct. So Ewald Hering, really in the late 1800s, I have early 1900s, but my memory is the late 1800s, suggested that there are 4 primary colors that are important. So remember, trichromatic theory of color vision is saying 3 color mechanisms, but he's saying 4 primary colors are important to us, and that they work in antagonistic pairs. And so I've talked about these as complementary colors before, because they're sometimes called that, but they're often talked about also as antagonistic colors or opposing colors because of these opposing responses that we see. And those are red, green, and then blue, yellow. That red and green are acting in an opposing manner. They're an antagonistic pair. And we need all 4 of those colors to really describe our experience of color vision. That's how I started talking about color vision, is that we needed to, in order to describe pretty much any color, we can use the words red and green and blue and yellow. And you'll notice that we never say it's a reddish green or a bluish yellow or a yellowish blue. That's not, we start to move towards green, and that's just not the way it works. And so that's some behavioral evidence, but we're going to see more. So a great deal of the behavioral evidence that Hering used was phenomenological evidence. And what I've just said was one piece, but also this color wheel is another piece. And you'll notice that those complementary colors, those opposing pairs, are across from each other on the color wheel. And what he noticed is if we start with red and we move over towards yellow, what we're doing each time is adding a little bit of yellow and a little bit of yellow and a little bit more yellow until we get to a light orange and then a yellow. At that point, we can start adding green, and we become a little bit more towards like a sea green, and then, a pea green. That's my sea greens over in the blue, but a pea green, and then we get to pure green with the absence of yellow. And then as we move towards the blue, we're adding a little bit of blue, then becoming a sea green, and then moving towards blue, which is, and we get to the left blue, and it's an absence of green. And then with that blue, we can start to add the red to go through some of those shades of purple. And then we get to red, which is an absence of blue, or an absence of blue or yellow, depending on which direction you're coming from. But his point was it makes sense from our phenomenological experience that there are 4 necessary colors, and they work in this kind of way. Later, in 1957, Hurwicz and Jameson also did some demonstrations, which my memory was that Hering did this as well, but Hurwicz and Jameson are given this credit, so I'm going to give it to them. These demonstrations that use after images. And so we're going to see why we're going to call this an opposing relation, even though Herring was arguing for that from this phenomenological evidence, it becomes even more clear with the afterimage demonstrations. Okay, do this demonstration. Do this demonstration. You have to pause this and stare at it for at least 45 seconds, looking at the fixation cross. Your eyes will have some slight movements, but if you're mostly looking at the fixation cross for 35, 40 seconds, I usually actually leave it on for a minute, but I'm talking now, and you should be looking at the fixation cross right now. Pause this, and then go to the next slide. Okay, if you paused that, what do you see now? Usually, this is a really fun, I like doing this in class because some people have seen it, a lot of people have seen it, but some people, I know who hasn't seen it because they do this, oh, wow. Okay, so that green, black, and yellow flag is now a red, white, and blue flag, right? And I usually ask, why do we see this? I'm gonna tell you why we see this. So the green has the opposing response or the antagonistic color of red. The black has the antagonistic opposing color of white, and the yellow has the antagonistic opposing color of blue. And so we have fatigued those neurons. And as we're going to see that we have those same kind of center surround receptive fields with excitatory and inhibitory processes going into some of our neurons down the line as we're getting this convergence of information. And so when we fatigue those neurons, we see the opposing color, the opposing response, in that kind of rebound that we see, if that makes sense. And as I stated with the experience of the red, white, and blue flag, that these are the three opponent mechanisms proposed by Herring. So black, white, red, green, and blue, yellow. And we can see here we have an excitatory, let's just use red, green, an excitatory red and an inhibitory green, but we're going to see that can go the opposite way, as I'm going to show some retinal ganglion cell activity from monkeys. So again, the physiological support for this came many, many years later, almost 100 years later. And they did single cell recording from retinal ganglion cells and from lateral geniculate nucleus cells. So that RG cells is retinal ganglion cells. Remember those from before. I know everybody wanted to come back to our center surround receptive fields. They're so fun. They're gonna haunt you. And then the lateral geniculate nucleus is the LGN there. Those cells provide physiological support for opponent neurons. And I'm just going to walk through the B plus yellow minus and just that one, even though you can see the same kind of response, but to red green in the R plus G minus, the red excitatory, green inhibitory. But the blue excitatory, yellow inhibitory, let's walk through that. If we look at just spontaneous activity, we see some neural firing. That's what each of those lines is showing, is the neural firing. If they show a 450 nanometer blue light, then we see what looks like excitatory activity, right? It's the neuron is firing faster. If we're at 510 nanometers, so a green light, it really looks about like spontaneous firing. I don't see a whole lot going on there. Might even be just a teeny bit of inhibition, but not really. At yellow, 580 nanometers, clearly being inhibited, right? There's almost no firing. That's the yellow inhibitory. Out at 660 nanometers, where we have a red light, it also looks pretty inhibitory, maybe not as strongly inhibitory as the yellow. And again, we see a similar kind of thing for red-green, except for the green being inhibitory and the red being excitatory. And here is a visualization given to us by Gouras in 1968 of those color opponent units in the retinal ganglion cells of monkeys. So we can have red excitatory, green inhibitory, where the red's in the center and the green's in the surround. Red inhibitory, green excitatory, again, red center, green surround. Green excitatory, red inhibitory, where the green is in the center and the red's in the surround, or green inhibitory and red excitatory, where the green's in the center and the red's in the surround. So we saw center surround receptive fields before. Here we see them again, but now adding color, and that the excitatory inhibitory relationship is between those opponent colors. And then we also show here a blue excitatory, yellow inhibitory. Some people suggest that this is the only one we have. Other people suggest we have all four of these possibilities with the blue and yellow. It just depends on the researchers you're talking to at this point. If we ask which of those theories is correct, I'm hoping the answer is very obvious now that it's both, right? Because really great behavioral evidence back before we could get the physiological evidence, and then years and years later they found physiological evidence, again, really clear physiological evidence for each of those color theories. And what's happening is both are correct. The trichromatic color theory is describing what is happening at the level of the cones, that we do have these three basic color mechanisms, as we have those three different kinds of receptors in the retina with their own visual pigment molecules that are going to then respond differently depending on the wavelength information. And then it's at a later level of processing that we see these opponent processes, where opponent process theory is more explaining what's going on after the convergence of information, and we get the integration of the inhibitory and excitatory signals. And yes, we get to talk about lateral inhibition again in a kind of different way now. But so we're seeing that, and we can measure that at the retinal ganglion cells as well as the cells in the lateral geniculate nucleus. We also actually see some opponent process neurons in the cortex as well, and we're going to save that for next.